Hey everyone, welcome to a deep dive. We're going to be talking about something really cool today. It's called protein engineering.
Yeah, we're going to get into the nitty-gritty of how scientists are actually designing and building proteins that you won't just stumble upon out in nature.
Okay, already I'm interested, but also a little intimidated. It sounds really complex.
It can be, but don't worry, we're going to break it all down for you. Think of it this way. Proteins are like the tiny machines that make life happen, right? They do all the heavy lifting in our bodies and all around us in nature.
Right, like each protein has its own special job to do.
Exactly. And protein engineering is like taking those natural machines and tweaking them, making them even better at their jobs, or even giving them entirely new jobs to do.
So we're talking about custom designed proteins, like tiny tools built for very specific purposes.
Exactly. Imagine proteins that could break down pollution or create new medicines or even be like tiny sensors that can detect harmful substances.
Oh, okay. That's some serious sci-fi stuff. But how does it actually work? Like, where do scientists even begin?
Well, there are two main approaches: rational design and directed evolution. It's kind of like choosing how to build a house. You could start with a super detailed blueprint and plan every step, or you could be more experimental and just see what cool stuff emerges.
I see. So rational design is like the blueprint approach, where scientists use their knowledge of like how proteins are structured and how they function to make really precise changes.
Exactly. They're basically acting like molecular architects, meticulously modifying those tiny protein building blocks.
Okay, that makes sense. But what if we don't have a complete blueprint? What if there are parts of the protein we don't fully understand?
That's where directed evolution comes in. It's more like mimicking how nature does things, that trial and error process, but in a much faster and more controlled environment. Scientists make a huge library of different protein variations with all sorts of random mutations, and then they see which ones actually do what they're looking for.
So it's like a massive audition for proteins, and only the best ones get the part.
Exactly. And the cool thing is, you don't need to know everything about the protein beforehand. You just need a way to test if it's doing its job.
Okay, I like that. So rational design is like you're a master craftsman meticulously sculpting something, but directed evolution is more like you're a breeder selecting for the traits you want.
I love that analogy. And just like in breeding, sometimes you get some really surprising and unexpected results.
Okay, so let's dig a little deeper into rational design. You mentioned that scientists need to understand the protein's structure and function. How do they actually figure that out?
Well, one way is using X-ray crystallography. It's like taking a molecular snapshot of the protein so you can actually see its 3D shape.
Wait, so you can actually see how the atoms are arranged in the protein?
Yep. And once they have that 3D structure, they can start to understand how different parts of the protein interact with each other and with other molecules.
I see. So it's like having a map of the protein's landscape showing you where to make changes.
Exactly. And there are other techniques too, like NMR spectroscopy, that also give us clues about the protein structure.
So scientists are using some pretty sophisticated tools to get a detailed picture of the protein they want to engineer.
They are. And once they have that picture, they can start making specific changes to the protein's amino acid sequence, which is like the chain of building blocks that makes up the protein.
Okay, so they're tweaking those building blocks to change the protein's properties or behavior.
Exactly. The goal might be to make it more stable or more active or even give it a whole new function.
It's mind-blowing that we can manipulate these tiny machines with that level of precision.
It really is. And rational design has led to some pretty amazing discoveries, like new drugs and enzymes with enhanced capabilities.
Okay, so rational design is all about precision and control, using our knowledge of the protein to guide the engineering process.
Exactly.
But what about directed evolution? How do scientists actually create all those different protein variations with random mutations?
One technique is called error-prone PCR. It's like deliberately introducing errors into the protein's DNA code. Those errors then lead to random changes in the amino acid sequence.
So it's like purposely making typos in the protein's instruction manual just to see what happens.
Yeah, pretty much.
Okay, that's a little scary, but I guess it makes sense.
And there's another technique called DNA shuffling, which involves mixing and matching DNA fragments from different versions of the protein.
So it's like taking the best parts from different recipes and trying to create a culinary masterpiece.
Exactly. We're introducing randomness, but with some strategy behind it.
I see. And once you have this whole library of mutant proteins, you have to find the winners, right?
Yeah.
The ones that actually have the trait you're looking for.
That's right. That's where screening comes in. It's like that big audition we were talking about.
Right. So what are some of the techniques scientists use to screen these proteins?
Well, one is called phage display, where you use viruses to display the different protein variations on their surface, and then you can see which ones bind best to a specific target molecule.
Whoa, using viruses to help us engineer proteins. That's wild.
Yeah, scientists are always coming up with creative ways to manipulate biological systems.
So with directed evolution, we're kind of letting nature do the heavy lifting, but we're guiding the process.
Exactly. It's led to some really cool discoveries, like enzymes that can break down plastic or even create new biofuels.
That's amazing. So we've talked about rational design and directed evolution. What else is there to explore in this protein engineering world?
Well, there's a technique called multiple sequence alignment, which helps us understand protein structure and function even when we don't have the full 3D picture. It's like comparing different regions of a historical text to try and figure out what the original message was.
Oh, that's interesting. So you're looking at similar proteins from different species to find clues.
Exactly. By comparing the amino acid sequences of related proteins, we can see which regions are highly conserved, meaning they haven't changed much over time. Those are probably really important for the protein's function.
It's like finding the parts of a machine that haven't changed much over the years. They must be pretty essential.
Precisely. And speaking of detective work, there's another technique called coevolutionary analysis.
Coevolutionary analysis. Okay, now you've got my attention. What is that?
This technique focuses on pairs of amino acids within a protein that mutate together over time. If one changes, the other tends to change too, so it's like they're linked somehow.
It's like finding two historical figures who always show up in the same stories. There must be a connection.
Exactly. And by analyzing these linked pairs, we can infer which amino acids are interacting with each other or are important for a shared function. It gives us clues about the protein's inner workings, even if we don't have the complete structural picture.
This is so cool. We're piecing together this puzzle using all these different techniques to figure out how proteins work and how we can engineer them.
Exactly. And once we have a good understanding of the protein's sequence and function, we can start to tackle the challenge of structural prediction.
Right, because knowing the amino acid sequence is one thing, but actually figuring out the protein's 3D shape is crucial, right? Shape determines function.
You got it. And while X-ray crystallography and NMR can give us those detailed 3D structures, sometimes those methods aren't possible for every protein. That's where computational methods come in.
Computational methods. So using computers to predict how the protein folds based on its sequence.
Exactly. It's a really tough problem, but scientists have developed some really clever tools for this. There are four main approaches, each with its own strengths and limitations. One is called ab initio modeling, which uses physics-based calculations to predict how the amino acid chain folds up.
So it's like trying to figure out how a string of beads will fold up based on the properties of the beads and the forces acting on them.
Pretty much. And then there's homology modeling, which relies on finding a protein with a known structure that's similar to the one we're interested in. And then we use that known structure as a starting point and adjust it based on the differences in the amino acid sequence.
So it's like finding a blueprint for a similar house and then modifying it to fit your needs.
Exactly. And there are two more approaches, fragment-based modeling and threading. They both involve comparing our protein to a database of known protein structures and trying to find the best match.
It's fascinating how each approach tackles this problem from a different angle.
It is, and they all rely on some pretty complex algorithms and a lot of computing power. But these tools are getting better all the time, giving us a much clearer picture of how proteins fold into those intricate 3D shapes.
Why is that so important?
Well, remember, shape determines function. By understanding the protein's 3D structure, we can learn a lot about how it interacts with other molecules, how it does its job, and how we can engineer it for different purposes.
Okay, so structural prediction is like getting that detailed blueprint, which is crucial for both understanding the protein's natural function and for designing new proteins.
That's it. And once we have a good understanding of the protein's structure, we can explore all sorts of cool engineering strategies like multivalent binding.
Multivalent binding, what in the world is that?
Imagine you're trying to hold on to a really slippery object with one hand. It might be hard to get a good grip. But what if you had multiple hands all grabbing the object at different points? It would be much harder for it to slip away, right?
Ah, I get it. So multivalent binding is like having multiple grabbing hands on a single molecule, making it stick much better.
Exactly. And this is really useful in designing new medicines. For example, There are bispecific antibodies that can target two different disease-related molecules at once, thanks to multivalent binding.
I see. So this isn't just theoretical stuff. It has real implications for how we treat diseases.
Absolutely. And multivalent binding is just one example of the cool strategies protein engineers are using.
Okay, my brain is officially full. We've covered so much already. From rational design to directed evolution, multiple sequence alignment, structural prediction, and multivalent binding. This field is huge.
You're telling me. And there's still so much more to explore, like the different types of directed evolution and the fascinating world of enzyme engineering.
I'm ready. Where do we go from here?
Welcome back.
It feels like we've covered so much ground already.
It's true we have, but there's still so much more to uncover. Last time we were talking about directed evolution. Remember that whole process of creating tons of protein variations and then seeing what cool traits emerge?
Right, like breeding dogs, but at a molecular level.
Oh,
So are there like different types of directed evolution, or is it like a one-size-fits-all kind of thing?
that's a great question. Actually, there are different categories within directed evolution, kind of like different breeding strategies. We can broadly group them into asexual and sexual methods.
Okay, asexual and sexual. So we're not talking about protein, getting romantic or anything.
Not exactly. But the terms do relate to how genetic material is handled. Asexual methods basically don't involve mixing genes from different parent proteins. Think of it like making copies of a blueprint, but each copy has slight random variations.
So like using that error-prone PCR technique we talked about,
Mhm.
where you intentionally introduce those typos into the DNA code.
Precisely. You're basically letting those mistakes play out and seeing if any of them lead to beneficial changes. Another asexual method is site saturation mutagenesis.
Okay, that sounds a bit more targeted. What's the strategy there?
With site saturation mutagenesis, you systematically change one specific amino acid position at a time, trying out every possible substitute to see what happens. It's a bit more controlled than just randomly making typos all over the place.
I see. So it's a more focused approach,
Yeah,
but still considered asexual because we're not mixing genes from different parents.
exactly. And then there are mutator strains, which are bacteria with a special talent for making errors when
Mhm.
they're copying DNA. They're like little mutation factories, churning out tons of variations for us to screen.
Wow, so they're like the typo kings and queens of the bacterial world.
You could say that. So what about the sexual methods? How do those work?
Okay, bring on the protein romance.
Well, just like in biological reproduction, sexual methods in directed evolution involve mixing genetic material from different parent proteins. It's like shuffling different versions of blueprints together, hoping to create something new and improved.
I like the way you think.
Yeah.
So we're taking bits and pieces from different successful proteins and combining them, like some kind of Frankenstein monster, but for good instead of evil.
Exactly. And one well-known technique is DNA shuffling, where you literally chop up and recombine different versions of the gene. It's a bit like taking the best features from different cars and trying to build a supercar.
That's a great analogy. So it's not just about introducing random mutations. It's about strategically combining successful elements from different sources.
Precisely. There's also in vitro homologous recombination, which basically means mixing and matching similar DNA sequences in a test tube. We're getting pretty good at playing matchmaker with these genes.
So we've got our asexual methods like error-prone PCR and site saturation mutagenesis. And then we've got our sexual methods like DNA shuffling and in vitro homologous recombination. It's like a whole family tree of protein engineering techniques.
It is. And there's one more member of that family that I want to introduce you to. It's called phage-assisted continuous evolution, or PACE for short.
PACE. Okay, I'm intrigued. Tell me more.
This technique uses viruses, specifically these things called bacteriophages, to rapidly evolve the protein inside bacteria.
Wait a minute. Viruses helping us engineer proteins? That's a twist I didn't see coming.
I know, right? It just goes to show how creative researchers have to be in this field. They're constantly finding new ways to manipulate biological systems. So in PACE, the viruses carry the gene for the protein we want to evolve, and they're constantly replicating and mutating inside the bacteria.
It's like turbocharged evolution happening inside these tiny bacterial hosts.
Exactly. And because the viruses are replicating so quickly, we can achieve significant evolution in a matter of days.
That's incredible. It's like fast-forwarding through millions of years of evolution in the blink of an eye. So we've got asexual methods, sexual methods, and now this virus-powered evolution technique called PACE. What else is out there?
Well, now that we've covered those techniques, I want to shift gears a bit and talk about a whole different category of protein engineering approaches. They're called in vitro non-homologous recombination methods.
Okay, new term alert.
Yeah.
In vitro non-homologous recombination. Break that down for me. What's so special about this category?
Up until now, we've been talking about techniques that rely on homology, meaning similarity in DNA sequences. But what if we could engineer proteins based on structural similarities, even if their sequences are quite different?
So we're looking beyond just the letters of the genetic code and focusing on the overall shapes and structures of proteins.
Exactly. And that's the power of non-homologous recombination methods. They allow us to create these hybrid proteins with brand new functions by combining structural elements from different sources, even if those sources aren't related at the sequence level.
Okay, that sounds incredibly powerful. But how do we actually do that? How do we combine structural elements from different proteins?
One technique is called exon shuffling.
Exon shuffling. Okay, I vaguely remember what exons are. Remind me.
Remember that genes are made up of coding regions called exons interspersed with these non-coding regions called introns. It's like a recipe with instructions and then extra notes scribbled in between.
Right, the exons are the part that actually get translated into protein.
Exactly. So exon shuffling involves cutting and pasting different exons from various proteins, creating new combinations with potentially new functions.
So we're rearranging the building blocks of proteins, like playing with molecular Legos. What kind of exons do we shuffle, though? Is there a strategy there?
There is. We can combine exons from orthologous genes, which are genes in different species that evolve from a common ancestor. It's like comparing different versions of a software program across different operating systems. You might find some interesting variations that can be combined.
So we're taking advantage of those evolutionary relationships to create new combinations.
Precisely. We can also shuffle exons from paralogous genes, which are genes within the same species that arose from gene duplication. Imagine you have two copies of the same recipe, and you start swapping ingredients between them. You might end up with something quite different.
That's a delicious analogy. So we can mix and match within a species, or even across species.
Exactly. And we can even combine exons from non-homologous genes that have functionally related domains.
Okay, hold on a second. Functionally related domains. What are those?
Think of a protein as a multi-tool, like a Swiss Army knife. It might have different parts that perform different functions, a blade, a screwdriver, a corkscrew. These different parts are called domains, and they often have similar structures even if they're found in different proteins.
Ah, I see. So we're basically taking the screwdriver from one protein and attaching it to the blade of another protein, even if those proteins are completely unrelated.
You got it. And by doing so, we can create these hybrid proteins with entirely new capabilities.
This is mind-blowing. We're literally mixing and matching protein building blocks from all over the biological world.
And that's just the tip of the iceberg. There are many other techniques for in vitro non-homologous recombination, each with its own unique approach and applications.
Okay, I won't ask you to list them all,
Yeah.
but it paints a picture of a vast and creative toolkit for protein engineering. From rational design to directed evolution, and now this fascinating world of non-homologous recombination. It's like we have this whole orchestra of techniques to play with.
You could say that. And just like with any orchestra, the key is to choose the right instruments and techniques to create the desired symphony of protein function.
So we've gone from designing proteins like architects to mixing their genes like mad scientists to basically swapping parts like we're building with Legos.
Yeah.
It's amazing to think we have that much control over these tiny molecular machines.
It really is incredible. And all of these techniques are leading to some pretty remarkable applications out there in the real world.
Okay, you've mentioned applications a few times. I'm really curious to hear some specific examples. How is protein engineering actually changing things?
Well, let's start with medicine, because that's probably where most people first encounter the impact of protein engineering.
Makes sense. I've heard of these things called biopharmaceuticals, which are like drugs made from biological sources.
Yeah.
Are those a product of protein engineering?
Absolutely. You can thank protein engineering for a lot of the cutting-edge biopharmaceuticals that are hitting the market. Remember Riloncip, that drug we talked about earlier? That was developed through directed evolution to treat a rare inflammatory disease. That's just one example.
So instead of traditional chemical drugs, we're now seeing more and more treatments based on engineered proteins.
Exactly. And these biopharmaceuticals often have some pretty big advantages. They can be designed to target very specific molecules in the body, which can lead to fewer side effects compared to some traditional drugs.
That's a huge deal. It seems like protein engineering is giving us a whole new level of control over how we treat diseases.
It really is. And protein engineering isn't just revolutionizing treatment, it's also transforming diagnostics, our ability to detect diseases early on.
Oh yeah, I've heard about these biosensors that can detect diseases. Are those based on engineered proteins too?
You bet. Imagine a tiny sensor that can detect minute traces of a specific biomarker in your blood or urine. That could give doctors an early warning sign for diseases like cancer.
Wow, that would be incredible. Catching a disease early can make all the difference. It's like having a microscopic detective on the lookout for trouble.
And the applications of protein engineering go way beyond medicine.
Okay, bring it on. Where else is protein engineering making waves?
Well, think about industries that rely on enzymes, those biological catalysts that speed up chemical reactions.
Right, they're like the workhorses of biology, breaking down food-building complex molecules.
Exactly. And by engineering enzymes, we can make them more efficient, more stable, or even give them entirely new capabilities.
So it's like giving those workhorses a supercharge.
Precisely. And this is revolutionizing all sorts of industries, like food processing, biofuel production, even laundry detergent manufacturing.
Hold on. Engineered enzymes in laundry detergent.
Okay.
You're kidding, right?
Nope. It's true. Protein engineering isn't just about high-tech medical breakthroughs. It's also about improving everyday products. Imagine enzymes that can break down stains at lower temperatures, saving energy and water. That's the power of protein engineering.
Okay, you've officially blown my mind. So we're talking about a wide range of applications, from life-saving medicines to more efficient and eco-friendly products. This field is truly remarkable.
It is, and that's just the beginning. Protein engineering is also being used to develop new materials with unique properties, biosensors, for environmental monitoring, bioremediation strategies to clean up pollution. We're talking about a field with the potential to address some of the world's most pressing challenges.
It's incredible to think that we can manipulate these tiny molecular
Right.
machines to solve such big problems.
And that's what makes this field so exciting. There's a sense of endless possibility.
I'm definitely feeling inspired after learning about all of this. It's amazing to see the impact that protein engineering is already having, and it sounds like we're only just getting started.
We are. And who knows, maybe some of you listening will be the ones to make the next big breakthrough.
That's a great note to end on. So to all you aspiring protein engineers out there, keep exploring, keep learning, and who knows, maybe you'll be designing the next generation of protein marvels. And that's a wrap for our deep dive into protein engineering. Thanks for joining us.